Hi everyone and welcome to Module 2. This week our focus is on the topic of hematology and chemistry and essentially some of the tests that go along with that. So I'm going to spend some time talking about the 3 tests that we'll focus on in the lab this week, and hopefully that'll help you get a little bit better of an understanding so when you come to class next week you are ready. So let's go ahead and get started. We're going to start with the topic of a hemoglobin. Now, hemoglobin is a vital protein molecule found in your red blood cells that has 2 primary functions. The first function is to carry oxygen from the lungs to the cells of the body. When the hemoglobin is carrying oxygen, it's called oxyhemoglobin. And this is where arterial blood is, because arterial blood has a high concentration of oxyhemoglobin, which gives it that bright red color. The second function of hemoglobin is to carry carbon dioxide, a waste product, from throughout the body back to the lungs, where it can be expelled through exhalation. Hemoglobin that is carrying carbon dioxide is called carboxyhemoglobin. And venous blood is darker in color than the arterial blood because it does carry that carboxyhemoglobin. Hemoglobin also consists of iron, which is where it gets its name, heme, and protein, which is where it gets that globulin. So hemoglobin is essentially iron protein. A molecule of hemoglobin contains 4 atoms of iron and an atom of oxygen, and they will attach each of the iron atoms to one another. Because of its ability to bind with oxygen, hemoglobin in the blood is responsible for carrying oxygen throughout the body. Thus, a higher hemoglobin level means that your body is able to transport more oxygen, and a lower level means that it can carry less oxygen in circulation. A low hemoglobin may indicate an iron deficiency anemia, where elevated readings are present in patients with polycythemia, and in extreme situations, maybe someone such as having burns would have a high level. Now, a normal level for adult females is 12 to 16 grams per deciliter. For males, it's 14 to 18 grams per deciliter. Hemoglobin can be measured either by an automated blood analyzer or a manual hemoglobinometer. In the lab, we will be learning how to use the manual hemoglobinometers. The manual method is less accurate, but it still can be done. It's just easier to draw it in a tube and do a CBC or even do a red blood cell count to get that information. I do want you to review procedure 47-2 in your book. Read through that before you come to class to give you a better understanding of how to perform a hemoglobin. Next, we have a hematocrit, and this will test the percentage of packed red blood cells in the total volume of blood. In order to do this, a blood sample is collected in a tube, then placed into a centrifuge, which will spin for about 15 minutes. This process separates the blood components into 3 layers. The red blood cells will sink to the bottom, the plasma will float to the top, and the buffy coat, which is a thin layer of buff-colored content, will actually separate the two. It mostly contains white blood cells and platelets. When we do this in the lab, we will be doing this by a capillary puncture. So you'll actually collect 2 tubes because you will compare them against one another. A normal hematocrit level is 40 to 50% in males and 35 to 45% in females. A low hematocrit may indicate anemia or a hemorrhage, and an elevated hematocrit may indicate dehydration or polycythemia. High glucose levels may falsely indicate elevated hematocrit levels also, so that's something we need to pay attention to. All right, and the last one that we are going to talk about today is the erythrocyte sedimentation rate, or also an ESR. Now, a hemoglobin and hematocrit are part of a CBC, or a complete blood count, but an ESR is not. You may hear it called an ESR, or you might hear it called as a SED rate. Now, in this, it also evaluates the red blood cells, but more specifically, it determines the rate at which red blood cells will settle at the bottom of a tube. When drawing a patient's ESR, this can be done by either using a Winthrop or Westergram method. The normal ESR for an adult female is 0 to 20 millimeters per hour, and in an adult male, it's 0 to 9 millimeters per hour. Now, with all of these, there is a procedure in the book that will relate. Again, with all of them, I highly suggest that you guys read them, go over the procedure in the book, because that will help you be prepared for the lab class. We'll spend the morning doing all of our assessments and things of this nature, and then we'll spend the afternoon doing your weekly skills checkoff. So sort of rooming, kind of learning how you would respond to providers, getting orders from providers, doing injections, doing blood draws, and then we'll also work on taking CMA exam preps. So it's something that we'll do each week just to get you guys prepared and ready for your CMA exam, because that is coming up quickly. All right, guys, if you have any questions or concerns, always please make sure that you are reaching out to me. You can reach me via email in the course messaging, or you can reach me by my direct line to the Rockford campus. You can find that in the upper right-hand corner of your course homepage. If nothing else, then I look forward to seeing you all Tuesday morning. Have a wonderful week, guys. Bye-bye.